Welcome to the Deep Dive. We're here to break down complex topics, making them easier to grasp.
Today, we're tackling a really fundamental area, sensory physiology, how we actually experience the world around us.
Exactly. We're using Chapter 7 of Vander's Human Physiology as our guide, our mission, to walk you through the core ideas, the mechanisms, the definitions, all in order, nice and clear.
Think of it like getting the key points of a lecture, but hopefully a bit more engaging. We'll decode the jargon.
We'll start with the big picture, those general rules that apply to all our senses, how stuff like light or pressure becomes, electrical signals.
And then we'll get into the specific vision, hearing, touch, taste, smell,
Yeah.
the whole lineup.
Ready to connect those dots?
Let's do it. So first up, general principles. Every sensory system fundamentally has three parts. You've got sensory receptors detecting things,
The detectors,
right? Then neural pathways to carry the message.
the wiring.
Exactly. And finally, parts of the brain that actually process that information.
Okay, and you mentioned processing. It's important to sort out sensory information, sensation, and perception, isn't it? They sound similar, but are quite different.
Absolutely crucial. Sensory information is just the raw data your system picks up. Could be blood pressure changes, stuff you're not even aware of.
I'm just monitoring.
Precisely. Now, if that info makes it to your conscious awareness, that's a sensation, like you feel a pain.
Okay, pausing me.
Right. But perception is the next step. It's your brain interpreting that sensation. Not just feeling pain, but knowing which tooth hurts and maybe why. It's the brain making sense of it all.
Giving it meaning. So how does that external thing, light or sound or whatever, become something our nerves can even work with?
That's the magic of sensory transduction. It's the process of converting that stimulus, energy, light, pressure chemical into electrical signals.
Yeah, the language of the nervous system.
Exactly. It starts as a graded potential at the receptor. If it's strong enough, boom, action potentials fire off in the afferent neuron.
And that pattern of firing isn't random, right? It's like a code.
It is a code. It tells the brain about the stimulus, how strong it is, where it is, what type it is, all packed into that electrical pattern.
And this whole process starts with the sensory receptors themselves. What are they exactly?
They're specialized structures, usually at the very ends of afferent neurons. Could be neuron endings, sometimes separate cells that talk to the neuron.
And they're waiting for a specific trigger.
Yes, the stimulus, that's the energy or chemical that turns them on. Each receptor type has what we call an adequate stimulus, the specific kind of input it's most sensitive to in normal life.
Like light for the eye receptors.
Perfect example. And they can be incredibly sensitive. We're talking detecting just a few molecules for smell or even a single photon of light.
Wow. Okay, and we group these receptors based on their adequate stimulus.
We do. The main classes are mechanoreceptors for physical things like pressure, touch, stretch.
Makes sense. Mechanical forces.
Thermoreceptors for temperature, hot and cold.
Got it.
Photoreceptors, obviously for light. Chemoreceptors, which respond to specific chemicals, think taste and smell, but also internal things like oxygen levels. Okay. And finally, nociceptors. These detect pain, signaling potential, or actual tissue damage from really intense stimuli. Could be extreme heat, pressure, or chemicals.
Pain receptors. Got it. So the stimulus hits the receptor. What happens electrically right then?
It causes ion channels in the receptor membrane to open or close. This changes the flow of ions, creating a receptor potential.
Which is a graded potential, right? Not an all-or-nothing action potential yet.
Exactly. It's graded. Its size depends on the stimulus strength. If this receptor potential is big enough to reach threshold.
Then it triggers the action potentials.
Yes, usually at the first node of Vander on the afferent neuron. And these action potentials then travel all the way to the central nervous system.
And the strength of the stimulus is coded how? Does the action potential get bigger?
Good question. No, action potentials are all or nothing. Their size doesn't change. Instead, a stronger stimulus creates a larger receptor potential, which in turn triggers a higher frequency, more frequent action potentials.
More spikes per second means stronger signal.
That's the core idea. It also affects how much neurotransmitter gets released at the synapse.
Okay, that leads nicely into adaptation. It's how we, stop feeling the chair we're sitting on after a minute, right?
Exactly. Adaptation is when a receptor decreases its sensitivity, fires less frequently, even if the stimulus is still there, unchanging.
Like tuning out the constant background noise?
Sort of. We have slowly adapting or tonic receptors. They keep firing maybe at a lower rate for the whole duration. Good for things you need to monitor constantly, like your body position.
Makes sense. And the opposite?
Rapidly adapting or phasic receptors. These guys fire strongly when the stimulus starts, maybe again when it stops, but they shut up quickly during the steady part. Excellent for detecting changes, movement, vibration.
Like feeling your phone vibrate but not noticing it just sitting in your pocket.
Perfect analogy. They give those on-off responses.
So clever. Okay, the signals are generated, they're adapting. How does the brain actually figure out what's going on from this stream of action potentials, the coding part?
Right. Primary sensory coding. This is all about converting that raw stimulus energy into useful information. What type it is, how strong it is, and where it's coming from.
Let's break that down. How does the brain know where the signal originated?
It starts with a sensory unit that's a single afferent neuron, plus all its receptor endings. Each unit has a receptive field.
The patch of skin or area of the retina or whatever that activates that specific neuron.
Exactly. And these fields often overlap, which is important.
Okay, we'll come back to overlap first. How does the brain know if it's heat or pressure or light? The modality.
Modality is coded by which receptors are activated. Each receptor type is tuned to its adequate stimulus and uses specific transduction mechanisms. So the pathway activated tells the brain the type. We sometimes talk about submodalities too, like cold versus warm or salty versus sweet.
And because receptive fields for different modalities can overlap, like you said.
You can feel multiple things at once from the same spot, like that ice cube feeling both cold and like pressure.
Gotcha. Now, intensity. We said stronger stimulus means higher frequency firing.
Right, that's one way. Action potential frequency in a single neuron goes up.
But there's another way.
Yes, recruitment. A stronger stimulus usually affects a larger physical area, so it activates more sensory units, recruiting neighboring neurons into action.
So higher frequency and more neurons firing signals greater intensity. Makes sense.
It does. Now, back to location. How precise can we be? This is acuity.
Like telling 2 points apart on your fingertip versus your back?
Exactly. The brain knows where, based on labeled lines, the idea that each pathway from a specific location and receptor type leads to a specific spot in the brain.
A dedicated line.
Sort of. Acuity depends on a few things. The size of the receptive fields, smaller fields mean better acuity.
Fingertips again.
Right. Also, the density of sensory units, more units packed in means finer detail. And how much the pathways converge, less convergence preserves more spatial information.
Okay, but you said overlap helps. How does that work?
It seems counterintuitive, but yeah. Imagine a stimulus hits an area. The neuron whose field is right in the center fires the most because receptor density is usually highest there. The overlapping neurons on the edges fire less strongly.
Ah, so the pattern of firing across overlapping fields gives the brain more info to pinpoint the center.
Precisely. And it gets even smarter with lateral inhibition.
This sounds important.
It really is, especially for vision and touch. The neuron most strongly activated actually inhibits the signals from its less activated neighbors on the edges.
So it dials down the fuzziness at the borders.
Exactly. It enhances the contrast between the center and the periphery, making the sensation much sharper and easier to localize. Think of that pencil tip on your finger. Lateral inhibition makes it feel like a distinct point.
Which explains why some sensations, like maybe pain or temperature, feel more diffuse. They use less lateral inhibition.
That's a big part of it, yes. Pain localization is often much poorer.
Fascinating. So these signals aren't just passively relayed, they're actively shaped on the way up.
Absolutely. That's central control of afferent information. Signals get modified at synapses all along the pathways. Inhibition can come from nearby ascending neurons, like lateral inhibition, or even from descending pathways coming down from the brain.
So the brain can essentially turn the volume up or down on incoming signals.
It provides flexibility. Think about focusing attention, or how pain can be modulated by mood or distraction. The brain gets a highly processed, curated version of the initial signal.
Okay, so let's talk about these pathways. How do the signals physically get from the periphery up to the brain?
Through ascending neural pathways. These are typically chains of 3 or more neurons synapsing with each other, running in parallel, bundles up the spinal cord and brainstem.
One neuron talks to the next, which talks to the next.
Right. And there's processing along the way. Signals can diverge, one neuron affecting many, or converge, many affecting one.
Are all pathways the same?
No, there's a key distinction. Specific ascending pathways carry info about a single modality, like touch or vision or hearing, to dedicated primary sensory areas in the cerebral cortex.
Like the somatosensory cortex for body senses, visual cortex for sight, auditory cortex for hearing.
Exactly. Taste goes to the gustatory cortex, smell to the olfactory cortex. And crucially, most of these specific pathways cross over somewhere along the way.
So left side of the body generally sends signals to the right side of the brain and vice versa.
That's the general rule. The olfactory system is a bit unique. Some pathways go straight to the cortex and limbic system without hitting the thalamus first.
Interesting. What's the other type of pathway?
Non-specific ascending pathways. These get input from multiple different types of sensory units. They don't tell you what specifically happened, but more signal general arousal or awareness like something's going on. They often end up in the brainstem reticular formation or less specific parts of the thalamus and cortex.
Okay, so the signal arrives at its primary cortical area. Is that the end of the story for perception?
Not at all. Processing continues in cortical association areas, regions right next to the primary zones.
This is where the real interpretation happens.
Yes, progressively more complex analysis, integrating information from different senses, linking it with memory, attention, language.
Like knowing a tree is upright, even if you tilt your head, combining vision and maybe vestibular input.
Perfect example. These areas also connect heavily with the frontal lobes and the limbic system, which adds emotional context and motivational significance to whatever you're sensing.
Which reinforces the idea that perception isn't just a raw feed of the outside world.
Definitely not. We've seen how receptor adaptation and pathway processing shape the signal. But internal things matter too. Your emotions, personality, past experiences all color how you perceive something compared to someone else.
And lots of sensory info never even reaches consciousness, right? Like blood pressure?
Right, it's filtered out or used for subconscious regulation. Plus, we just don't have receptors for everything out there, like certain types of radiation.
And sometimes the system can generate perception without external input, like phantom limb.
Exactly. That shows how cortical areas can rewire and generate sensations even when the original input source is gone. Drugs or mental illness can also dramatically alter perception. It's really a blend of the stimulus, the neural processing, and the brain's interpretation.
Incredible complexity. Okay, let's shift gears and apply these principles to the specific senses. How about we start with somatic sensation? Touch, temperature, pain, that group.
Sounds good. Somatic sensation covers feelings from your skin, muscles, bones, joints. Think touch, pressure, proprioception, body position movement, temperature, pain, and itch. Uses various somatic receptors.
Let's take touch and pressure.
These rely on different types of mechanoreceptors in the skin, often specialized neuron endings may be encapsulated. They basically translate physical deformation into ion channel activity. Some adapt rapidly good for feeling movement or vibration. Others adapt slowly better for sustained pressure.
And their receptive fields vary, too.
Yep. Small, well-defined fields on fingertips give you precise detail. Larger, fuzzier fields elsewhere might signal skin stretch or joint movement.
What about knowing where your limbs are? Proprioception.
Key players here are muscle spindle stretch receptors, detecting muscle length and changes, and Golgi's tendon organs, monitoring muscle tension. But vision and your vestibular system also play crucial roles. Kinesthesia is the specific term for sensing joint movement.
Okay.
Detected by thermoreceptors, which are mostly free neuron endings. Yeah. The actual sensors are proteins called transient receptor potential TRP channels.
TRP channels?
Yeah. Different versions of these ion channels open up at different temperature ranges. When they open, ions flow in, depolarizing the neurons.
Is that why chili peppers feel hot and mint feels cool?
Exactly. Capsaicin in chili and menthol activate specific TRP channels, usually associated with heat and cold, respectively. It's a chemical tricking the temperature sensor.
Clever. Now, the unpleasant ones, pain and itch.
Pain is detected by nociceptors, also free nerve endings. They respond to intense stimuli, mechanical, thermal, or chemical, like substances released from damaged cells, histamine, bradykinin, H+ ions.
And the signals involve specific neurotransmitters.
Yes, glutamate and particularly substance P are important in pain pathways.
What about referred pain, like feeling heart attack pain in your arm?
That happens because sensory nerves from internal organs often converge onto the same spinal cord neurons that receive input from skin areas. The brain gets confused about the origin and attributes the visceral pain to the more common somatic source.
Crosstalk in the wiring. And hyperalgesia.
That's increased pain sensitivity after an injury, like how a sunburn hurts more later. It highlights that pain isn't just about the stimulus, it's heavily modulated by past experience, emotions, inflammation.
Can we actively reduce pain, analgesia?
Yes, the body has its own pain control systems. Descending pathways from the brainstem can inhibit pain signals in the spinal cord, often using endogenous opioids or natural morphine-like substances.
Is that how things like acupuncture or maybe even the placebo effect might work?
That's the leading theory. They activate these descending opioid-releasing pathways. Another method is transcutaneous electrical nerve stimulation, TENS, where stimulating large touch fibers seems to inhibit the transmission from smaller pain fibers, like rubbing a bumped elbow.
Cake control theory, sort of. And itch.
Itch is considered a separate sensation, though it shares some pathways and mechanisms with pain. It can be triggered mechanically or chemically, like by histamine.
Okay, how do all these somatic signals, touch, pain, temperature, get to the brain cortex?
Two main highways. First, the anterolateral pathway, sometimes called spinothalamic. It handles primarily pain and temperature. Neurons synapse quickly in the spinal cord, cross over to the opposite side almost immediately, and then head up to the thalamus and cortex.
Crosses low down. What's the other one?
The dorsal column pathway. This carries mainly fine touch, pressure, and proprioception. It ascends on the same side of the spinal cord it entered, synapses first in the brainstem, then crosses over to the opposite side to reach the thalamus and cortex.
So they cross at different levels, but both end up sending info from one side of the body to the opposite cerebral hemisphere.
Correct. And the somatosensory cortex itself has that famous map of the body, the homunculus, where areas with more sensory input, like fingers and lips, get disproportionately large cortical representation.
And you mentioned this map can change, like with phantom limbs.
Exactly. It's dynamic.
Yeah.
If input from a limb is lost, cortical areas nearby, say for the face, can actually expand into that silent limb cortex. Stimulating the face might then trigger sensations in the phantom limb because the wiring has reorganized.
Truly remarkable brain plasticity. Let's switch senses now. How about vision?
Okay, vision. The stimulus is light, which is just a tiny sliver of the electromagnetic spectrum, roughly 400 to 750 nanometers in wavelength.
And wavelength determines the color we perceive.
That's right. Shorter wavelengths are blues, violets, longer are reds.
Can you give us a quick tour of the eye's structure?
Sure. Think of 3 layers. The tough white outer layer is the sclera, which becomes the transparent cornea at the very front. The cornea does most of the light bending, the initial focusing.
Layer 1.
Layer 2, underneath, is the choroid. It's pigmented, helps absorb stray light. Up front, it specializes into the iris, the colored part that controls the pupil size, and the ciliary muscle, which connects via zonular fibers to the lens.
So the iris controls the pupil, the opening for light, and the ciliary muscle controls
the lens, which sits just behind the iris. It's crystalline and does the fine-tuning of focus.
Got it. And the innermost layer?
That's the retina. It's actually an extension of the brain, packed with photoreceptors and other neurons.
Where the light detection happens. Any key spots on the retina?
Yes, the macula lutea is the central area, and within it is the fovea centralis, a small pit where vision is sharpest because it's densely packed with cones. There's also the optic disc, where the optic nerve exits. No receptors there, so it creates our blind spot.
Okay, focusing. The cornea does the main bending. The lens fine-tunes. How does the lens change shape?
Through accommodation. The ciliary muscle controls it. When it contracts, the tension on the zonular fibers slackens, and the lens naturally becomes more rounded, more curved, which increases its bending power for focusing on near objects.
Contract muscle near vision.
Right. When the ciliary muscle relaxes, the zonular fibers tighten, pulling the lens flatter, which is better for distant vision.
Relax muscle equals far vision. What happens when this gets harder with age?
That's presbyopia. The lens loses elasticity, so it doesn't round up as easily for near vision, hence reading glasses.
And common issues like nearsightedness or farsightedness?
Myopia or nearsightedness usually means the eyeball is too long, or the lens cornea bends light too much. Distant objects focus in front of the retina, corrected with a concave lens.
And hyperopia?
Farsightedness. Eyeball too short, or not enough bending power. Near objects try to focus behind the retina, corrected with a convex lens. The pupil also adjusts, constricting in bright light or for near vision to sharpen focus, dilating in dim light.
All right, let's go inside the retina to the photoreceptors. Rods and cones. How do they actually turn light into a signal?
Rods are for dim light, black and white vision. Cones are for brighter light and color vision. Both have an outer segment with stacks of discs containing photopigments. These are proteins called opsins attached to retinal, which is derived from vitamin A and is the light-absorbing part.
And the process is unusual. They're active in the dark.
It's unique, yeah. In the dark, photoreceptors are actually depolarized. High levels of a molecule called cGMP keep certain cation channels open. letting Na plus and She2 plus flow in.
Depolarized in the dark. So what happens when light hits?
Light hits the retinal molecule, causing it to change shape and detach from the opsin. This activates a G protein called transducin.
G protein cascade.
Exactly. Transducin activates an enzyme that breaks down cGMP, so cGMP levels fall, the cation channels close, ion influx stops, and the cell hyperpolarizes.
Light causes hyperpolarization. That's the signal.
That's the signal that gets passed on.
And this links to adapting to light levels.
Yes, dark adaptation, moving from bright to dark. Your rods were bleached, retinal detached from opsin. It takes time for retinal and opsin and rods, to recombine, making rods sensitive again. Needs vitamin A.
Explains why it takes a few minutes to see in a dark room.
And light adaptation, going from dark to bright. Initially blinding, as supersensitive rods get overwhelmed, they quickly bleach out, becoming unresponsive, letting the less sensitive cones take over for sharp color vision in bright light.
Okay, signal generated. How does it get from the photoreceptor to the brain?
Photoreceptors synapse onto bipolar cells. Bipolar cells then synapse onto ganglion cells. Importantly, only the ganglion cells fire action potentials.
Bipolar cells are intermediaries.
Yes, and there are different types. We have on-end pathways that get excited by light hitting the photoreceptor, which hyperpolarizes, and OAF pathways that get excited when the light goes off, photoreceptor depolarizes. This split processing enhances contrast and edges.
Complex processing right there in the retina.
Absolutely. Horizontal cells and amacrine cells also provide lateral connections for further processing. Then, the axons of all the ganglion cells bundle together to form the optic nerve, cranial nerve 2.
Which leaves the eye at the blind spot. Where does it go next?
The two optic nerves meet at the optic chiasm. Here, fibers from the nasal inner half of each retina cross over to the opposite side. Fibers from the temporal outer half stay on the same side.
So information from the left visual field, seen by the right half of each eye, goes to the right brain, and vice versa.
Exactly. This crossing ensures that each hemisphere gets input from both eyes, which is essential for binocular vision and depth perception. After the chiasm, the fibers continue as optic tracts.
And their main target?
Mostly the lateral geniculate nucleus, LGN, specific part of the thalamus. The LGN keeps info about color, form, movement somewhat segregated and relays it to the primary visual cortex in the occipital lobe.
Where conscious perception of vision begins.
Right. Though information also goes elsewhere, like to the suprachiasmatic nucleus for regulating circadian rhythms via special melanopsin-containing ganglion cells, and to the brainstem for controlling eye movements.
Let's quickly touch on color vision. How do the cones manage that?
We have three main types of cones, often called L, red-sensitive, M, green-sensitive, and S, blue-sensitive, based on the wavelengths they respond to best. Your perception of any color depends on the relative levels of activity across these three cone types.
Like mixing primary colors?
Kind of. The brain compares the output. There are also opponent color cells further down the line, like ganglion cells, that are excited by one color range, say red, and inhibited by another, say green. This sharpens color distinctions.
And color blindness.
Most commonly red-green color blindness. It's usually genetic, linked to the X chromosome, so more common in men. They lack or have faulty red or green cone photopigments, making it hard to distinguish between those shades.
Okay, and briefly, eye movements.
Controlled by 6 muscles attached to each eyeball. We make fast, jerky saccades to shift gaze quickly, scan scenes, and prevent adaptation. And slow movements for tracking moving objects or stabilizing gaze when our head moves, often driven by vestibular input.
Got it. Any common eye diseases we should mention?
Cataracts are very common, clouding of the lens, usually age-related, fixed surgically. Glaucoma is serious damage to the optic nerve, often due to high pressure inside the eye, can cause irreversible blindness if untreated. And macular degeneration, AMD, affects the maculofovea, causing loss of central sharp vision, also increases with age.
All right, a lot going on in vision. Let's move to audition hearing. The stimulus here is sound waves.
Yes, sound travels as pressure waves through a medium, like air. Zones of higher pressure, compression, alternate with lower pressure, rarefaction. Needs molecules to vibrate, no sound in a vacuum.
And the properties of the wave relate to what we hear.
Right. The amplitude of the pressure wave relates to loudness, measured in decibels, EV. The frequency of the waves determines the pitch, measured in hertz. Humans typically hear from 20 hertz to 20,000 hertz.
What about the quality of a sound? Like a trumpet versus a violin playing the same note?
That's timbre. It comes from the presence of overtones, additional frequencies mixed in with the fundamental pitch.
Okay, how does the ear capture and process these waves? Start with the outer ear.
The pinna, the visible part, and the external auditory canal collect sound waves, and funnel them towards the tympanic membrane, or eardrum.
The eardrum vibrates, then what?
The vibrations pass into the air-filled middle ear. Here, three tiny bones, the ossicles malleus, ink, and stakes, form a lever system. They transmit vibrations from the large eardrum to the much smaller oval window, which is the entrance to the inner ear.
Why the bones? Just passing it along.
They actually amplify the pressure significantly, maybe 15–20 times. This is crucial because the inner ear is fluid-filled, and it takes more force to move fluid than air.
Overcoming the impedance mismatch, what about the eustachian tube?
That connects the middle ear to the back of the throat, pharynx. It opens when you swallow or yawn to equalize the air pressure on both sides of the eardrum. Prevents discomfort or damage.
And those little muscles in the middle ear?
The tensor tympani and sopedius. They contract reflexively to loud sounds, dampening the ossicle vibrations to protect the inner ear. Also reduces the perceived loudness of your own voice.
Smart. Okay, into the fluid-filled inner ear, the cochlea.
The cochlea is spiral-shaped, like a snail shell. It contains the cochlear duct, which houses the actual sensory receptors. This duct is filled with a fluid called endolymph, which is unusually high in potassium, K+. The chambers above and below the duct contain paralymph, more like typical extracellular fluid.
So the stapes pushes on the oval window,
creating pressure waves in the paralymph of the upper chamber, scala vestibuli. These waves travel through the cochlea and cause the basilar membrane, the floor of the cochlear duct, to vibrate.
And does the whole membrane vibrate the same?
No, and this is key. The basilar membrane changes in stiffness and width along its length. The end closer to the oval window is narrow and stiff, vibrating most to high-frequency sounds. The far end is wider and more flexible, responding best to low-frequency sounds.
A frequency map laid out along the membrane.
Exactly. The pressure waves eventually dissipate at the round window, another small membrane-covered opening.
So where are the actual receptors that detect this vibration?
They sit on top of the basilar membrane in a structure called the organ of Corti. These are the hair cells, mechanoreceptors with tiny bristle-like structures called stereocilia on top.
Hair cells, how do they work?
There are inner hair cells, single row, primary detectors, and outer hair cells. Multiple rows seem to fine-tune the response. When the basilar membrane moves up and down, the hair cells move too. Their stereocilia bend against an overlying membrane called the tectorial membrane.
Bending the hairs is the trigger.
Yes. When stereocilia bend towards the tallest one, little protein filaments called tip links physically pull open mechanically gated ion channels at the tips of the stereocilia.
And because they're bathed in high K+ endolymph.
K+ flows into the cell. This is unusual. K+ influx causes depolarization here. Depolarization opens voltage-gated calcium channels, triggering the release of the neurotransmitter glutamate,
which excites the afferent neurons of the vestibulocochlear nerve, cranial nerve 8.
Precisely. The louder the sound, the more the basilar membrane vibrates, the more the stereocilia bend, the more glutamate is released, and the higher the frequency of action potentials in the nerve.
And loud sounds can damage these delicate hair cells.
Very easily. Damage is often permanent, leading to hearing loss. It can also cause tinnitus, that persistent ringing or buzzing sound.
Okay, auditory signals head up the nerve. Where to next?
First up is the brainstem, cochlear nuclei. Here, information from both ears starts to converge, which is important for figuring out where sounds are coming from, using differences in timing and intensity between the ears.
Sound localization.
Then signals relay through other brainstem nuclei up to the thalamus, and finally reach the primary auditory cortex in the temporal lobe.
Is there a map there, too?
Yes, the auditory cortex is tonotopically organized, mapped by pitch. Different neurons respond best to different frequencies, and also to more complex features like sound duration, loudness changes, or specific patterns. And like other senses, there are descending pathways allowing us to focus attention.
What about solutions for hearing loss?
Hearing aids basically amplify sounds entering the ear canal. For more severe damage, cochlear implants can bypass the damaged hair cells and directly stimulate the auditory nerve fibers electrically, providing a representation of sound.
Amazing technology. Now, the inner ear isn't just for hearing, right? There's the vestibular system, too.
Correct. Also housed in the inner ear, the vestibular system gives us our sense of balance and detects head motion and orientation relative to gravity. It uses hair cells, too, similar to the cochlea.
Oh, what are the main parts?
The vestibular apparatus consists of 3 semicircular canals and 2 chambers called the utricle and saccule, all filled with endolymph.
What do the semicircular canals detect?
They detect angular acceleration, rotational movements of the head, like nodding yes, shaking no, or tilting your head side to side. They're oriented in 3 perpendicular planes.
How do they sense rotation?
Each canal has a bulge called an ampulla, containing a gelatinous structure called the cupula. Air cell stereocilia are embedded in this cupula. When your head turns, the canal moves, but the endolymph inside lags behind due to inertia. This lagging fluid pushes the cupula, bending the stereocilia.
And bending signals the rotation.
Yes, specifically the change in rotation rate. If you spin at a constant speed, the fluid eventually catches up, the cupula returns to center, and the signal stops until you speed up or slow down.
What about the utricle and saccule?
These detect linear acceleration changes in straight-line motion, like speeding up in a car, and also the position of your head relative to gravity, head tilt.
How do they work?
Their hair cells have stereocilia embedded in a gelatinous layer containing tiny calcium carbonate crystals called otoliths, ear stones. These make the layer heavy.
So gravity or linear acceleration pulls on the heavy otolith layer.
Exactly. That pull bends the stereocilia, signaling the change in head position or linear motion.
What does the brain do with all this vestibular info?
Several crucial things. It's vital for controlling eye movements, to keep your gaze stable when your head moves, the vestibulo-ocular reflex.
So things don't look blurry when you walk.
Right. It's essential for posture and balance, coordinating muscle adjustments. It contributes to our conscious awareness of body position and movement in space.
And when it goes wrong or conflicts with other senses.
That can lead to dizziness, vertigo, or motion sickness, often caused by a mismatch between what your eyes see and what your vestibular system feels.
Where do the signals go?
From the vestibular hair cells via the vestibular branch of the vestibulocochlear nerve to vestibular nuclei in the brainstem. From there, info goes up to the thalamus, and then to vestibular centers in the cortex, parietal lobe, and also down the spinal cord for postural reflexes, and to eye muscle control centers.
Okay, last major category. The chemical senses, taste and smell.
Right, relying on chemoreceptors. These detect chemicals influencing appetite, digestion, and helping us avoid potentially harmful things.
Yeah.
Let's start with gustation, or taste.
Receptors are in taste buds.
Yes, about 10,000 of them, mostly on the tongue's lingual papillae, but also elsewhere in the mouth and throat. Each bud contains specialized taste receptor cells with microvilli to bind chemicals, supporting cells, and basal cells that replace worn-out receptor cells.
How are they activated?
Food molecules have to dissolve in saliva, then enter a taste pore to reach and bind to receptors on the microvilli.
And we have distinct taste categories.
Traditionally 5 basic ones: sweet, sugars, artificial sweeteners; sour acids, high H+ concentration; salty, primarily sodium ions and APO+; bitter, diverse group often associated with toxins' evolutionary protection; and umami, savory taste, triggered by glutamate, like in MSG.
Any others?
There's ongoing research suggesting maybe a distinct receptor for fatty acids exists too.
How do these different tastes trigger a signal?
The mechanisms vary. Salty is simple. Na+ ions directly enter channels, depolarizing the cell. Sour, H+ ions block K+ channels, which also leads to depolarization. Sweet, bitter, and umami generally involve G protein-coupled receptors linked to various intracellular signaling pathways that ultimately cause depolarization and neurotransmitter release.
And taste perception is more than just these 5.
It's hugely influenced by smell, but also texture, temperature, even pain, like spicy heat from capsaicin. Taste signals travel via cranial nerves to the gustatory cortex near the mouth area of the somatosensory map.
Okay, finally, olfaction smell. Hugely important for flavor.
Definitely. Explains why food seems bland when you have a cold. The receptors are olfactory receptor neurons, located high up in the nasal cavity in the olfactory epithelium.
These are actual neurons.
Yes, specialized afferent neurons. Their dendrites extend to the surface and have long cilia bathed in mucus. These cilia contain the odorant receptors. Uniquely, these neurons are regularly replaced throughout life by stem cells, about every 2 months.
How do we detect so many different smells?
Odorant molecules in the air dissolve in the mucus and bind to specific odorant receptors on the cilia. This activates a G protein pathway, increases CAMP, opens cation channels, and depolarizes the neuron, triggering action potentials.
Is it one receptor per smell?
No. Humans have around 400 different types of odorant receptor proteins. Each receptor neuron typically expresses only one type. But most odorant molecules can bind to multiple different receptor types, and each receptor type can bind multiple different odorants, though with varying affinities.
Ah, so it's the combination, the unique pattern of activation across different receptor types that the brain interprets as a specific smell.
Exactly. That combinatorial coding allows us to potentially discriminate thousands, maybe even trillions of different odors.
Where do these signals go? You mentioned it was unique.
Right. The axons of these neurons form the olfactory nerve, cranial nervi. They pass directly through bone into the olfactory bulbs on the undersurface of the frontal lobes. Significantly, this pathway bypasses the thalamus on its first relay to the cortex,
Straight to the olfactory cortex.
and also directly to parts of the limbic system, the brain's emotional centers. This direct link is why smells can trigger such strong emotions and memories.
That makes so much sense. What affects our sense of smell?
Lots of things. How attentive you are, if you're hungry, often more sensitive. Gender, women sometimes test better. Smoking decreases sensitivity. Age tends to decline. And obviously, the state of your nasal passages, like congestion from a cold. Lack of smell is called anosmia. So wrapping up, we've really journeyed through how our bodies sense the world, from the basic principles of turning stimuli into electrical codes,
Sensory transduction, coding intensity, and location.
to the amazing specializations of each sense. Vision's optics and photochemistry, hearing's mechanical amplification and frequency mapping, the body senses, balance, and the chemical detection of taste and smell.
And things like lateral inhibition really highlight how the signals are actively processed and refined, not just relayed.
Absolutely. The rich, detailed world you perceive is ultimately constructed by your brain from these patterns of electrical activity, constantly shaped by adaptation, central control, and even your own internal state. It's an incredible dynamic process.
It really is. Well, thanks for diving deep with us into Vander's Chapter 7 on sensory physiology. We hope this has given you a solid grasp and maybe a new appreciation for the systems connecting you to, well, everything.
From the entire Deep Dive team, thank you for joining us on this exploration. Keep that curiosity going.